Thank you for the kind introduction. My name is Taeyeong Kim from the Manufacturing and Instrumentation Lab in KAIST, Korea. I'd like to thank you for the opportunity to present at IEEE Sensors 2024. Today, I will be presenting on the development of a microfluidic platform for refractive index measurement of liquid. The measurement of liquid refractive index plays an important role in applications such as purity and concentration measurement of liquid and process monitoring of biological and chemical processes. For instance, in permeation manufacturing, it helps ensure the consistency of a solution by measuring the refractive index of liquid depends on concentration. In food industry, refractive index measurements are applied to determine the concentration of sugar solutions, which is vital in maintaining product quality. Additionally, refractive index measurements can also be applied to biological processes. This includes monitoring the refractive index of cells, which helps in understanding the composition and behavior of cell suspensions. Lastly, binary mixture identification relies on refractive index measurement to accurately determine the proportion of different substances in a mixture, such as organic solvent and water, which is important in chemical engineering processes. Liquid refractive index is typically measured using methods such as refractometers or interferometers. While these methods offer high accuracy, they require relatively large sample volumes and face challenges in detecting real-time change in refractive index in response to stimuli. Microfluidic approaches have emerged as an alternative for measuring liquid refractive index. Microfluidic devices offer several advantages, including the ability to handle small sample volumes, integrate multiple functions on a single chip, and provide real-time analysis. However, this approach often requires the implantation of waveguides within microchannels. The invasive nature of waveguide insertion complicates device fabrication. Additionally, precise alignment is necessary to specially fix the waveguides within the channel, which makes the setup more complex and restrict refractive index measurement to specific regions. So in this study, we present a platform that integrates near-infrared interferometer with a self-assembled cavity in silicon for the measurement of liquid refractive index. The self-assembled cavity featuring multiple parallel fluidic ports is fabricated. This cavity is then bonded to a PDMS interface, which includes one optical access hole and two tube connection holes. Using the near-eye interferometer, we perform a vertical piezo scan within the liquid field channel to capture the interference patterns. The distance between the first and second interference patterns remain constant as it is determined by the silicon thin film. However, the distance between second and third interference patterns varies depending on the liquid filling the cavity. From these patterns, we extract the refractive index by measuring the optical depth between interfaces at a specific point. So, as shown in the image, we develop a near-infrared interferometer. The setup is similar to an light microscope, and we use an interferometric objective lens with a piezo positioner to obtain scanned interferograms. During scanning, all the light reflected from the interfaces within the numerical aperture is corrected by the interferometric objective lens. When the optical path difference between the reference and sample beams is smaller than the coherence length, interference patterns are obtained for each interface, allowing the reflected light from each layer to be distinguished. The relative position of each layer is determined by the interference maximum. Since the optical distance is the product of the channel height and real part of the refractive index, we can extract the real part of the refractive index using the known channel height. We fabricate the microchannels using silicon-on-nothing technology. Silicon-on-nothing refers to a monocrystalline silicon thin film suspended over an empty space, which is formed through surface diffusion when a silicon hole array is annealed at high temperatures. As shown in the image, initially individual vertical cavities are formed, and as the annealing progresses, these cavities merge to form horizontal cavities. To inject liquid into the microchannel, we positioned a larger 4 micrometer hole array around the smaller 1 micrometer hole array, which forms the silicon nesting structure through surface diffusion. During high temperature annealing, the smaller holes form the channels, while the larger holes remain open because their surface diffusion rate is much slower, preventing the surface from closing. In our previous work, we optimized the diameter and spacing of these larger holes to ensure proper coupling. Since the microchannel is connected to the larger holes, they can be used as the fluid inlet and outlet. We connected fluid ports to the microchannel using a PDMS interface through plasma bonding. To control the flow within the system, we use two electronic pressure regulators that apply differential pressure for pressure-driven flow throughout the entire fluidic setup. The amount of liquid required for the measurement is only in the tens of nanoliters. The bottom of the silicon wafer is maintained at 20 degrees Celsius using a thermoelectric module. The microchannel with a PDMS interface is placed under a near-ion interferometer, allowing us to measure the refractive index of the liquid inside. To compare with our measurement, we conducted optical simulations for the interferometer using FDTD in ANSYS numerical software. Both simulations and experiments utilize the near-infrared wavelength range. In the simulations, a pulse with same spectral configuration as the continuous wave source used in the actual setup is emitted. The pulse reflected from the beam splitter is recorded as the first signal by the monitor, while the pulse reflected from the microchannel through the beam splitter is recorded as a second signal. These two signals are superimposed with a relative time difference and then integrated over time. The integration represents the intensity of the interferogram and the optical path length, which is the product of the speed of light and delta t. By repeating the superposition and time integration for different optical distance values in the 0 to 20 micrometer range, an interferogram signal as a function of piezo position is generated. Comparing the result when the self-assembled cavity is filled with air and versus DI water, we observed that the first and second interference peak near 4 micrometer and 50 micrometer remain unchanged. However, the larger refractive index of DI water increased the optical path length, shifting the third interference peak to the right. The optical path lengths between the second and third peak are the product of the cavity height and the refractive index of the material inside the cavity. The optical path length is measured using the near-ion interferometer when the cavity is filled with air and DI water. As the cavity is filled with DI water, the optical path length increases compared to when it's filled with air, causing the third interference pattern to shift to the right. However, the first and second interference patterns caused by the silicon membrane remain unchanged. When comparing the near-ion interferometer measurement of air and DI water in microchannel with the FETD calculated interferogram, we observe a notable trend of attenuated relative intensity between the peaks. This is because the FETD calculation assumes optically perfect flat surfaces, whereas the actual surface of the microchannel is not ideally smooth and contains periodic structures smaller than 100 nanometer. This non-negligible surface roughness induces diffraction, which leads to a reduction in peak intensity. Despite the difference in intensity, the optical distance between peaks remain consistent. We injected various liquids into the microchannel and measured their refractive indices. The measured refractive indices of these liquids differ by less than 0.4% from the values reported for liquids in the near-infrared range. This demonstrates that we can accurately measure the refractive index of the injected liquid. Additionally, we measure the refractive index of DI water as a function of sodium chloride concentration and room temperature below the saturation level. As shown in the graph at the bottom center, the third peak of the Larkin modulus shifted according to the varying NaCl concentrations, allowing us to measure the refractive index. The refractive index of NaCl solution at different concentrations exhibit a linear relationship with concentration, as shown in the graph, with a difference of less than 0.5% compared to reported values in the near IR range. Using the near eye interferometer, which collects data for each pixel through a CCD, we can capture the 2D distribution of leakage refractive index. For instance, we first fill the cavity with mineral oil, followed by DI water, with the injection proceeding in one direction from left to right. This sequential filling forms an interface between two immiscible leakage inside the cavity. The two regulators are used solely to precisely control the differential pressure. By measuring in a 30 micrometer by 30 micrometer area at the interface, we confirm that we can detect the boundary between the two immiscible leakage. The microchannel proposed in this study is made of silicon, making advantageous for temperature control. According to FA calculation, when the bottom temperature is of the wafer is set, the surface temperature remains within 1 Kelvin. Using a thermoelectric module at the bottom of the wafer, we measure the refractive index of leakage at different temperatures. We conducted measurement on 4 leakages in total and observed that the refractive index decreases as the temperature increases. For 3 leakages with previously reported refractive index data in the near infrared range, our measurement showed less than 5% difference across the temperature range. To conclude, we have developed a platform that integrates a self-assembled cavity in silicon with a custom-built near-infrared interferometer for liquid refractive index measurement. We fabricated a 2D hole array in silicon where the self-assembled cavity formation was achieved through high-temperature annealing. And our platform demonstrates highly accurate refractive index measurement with values within 0.4% of reported values for pure leakage samples. Additionally, we successfully detected the interface between immiscible leakages such as DI water and mineral oil by acquiring refractive index contours. Finally, we demonstrated accurate NaCl concentration determination in DI water using our interferometric measurement method. We also performed temperature-dependent refractive index measurement for various leakage samples. I'd like to express my gratitude to Professor Jungchul and researchers in the Manufacturing and Instrumentation Lab for their support. This research was funded by the National Research Foundation of Korea grant, supported by Korean government.